Okay, good morning, good afternoon, and good evening, depending on where you are. My name is Mohammed. I'm Cynobiologicals Marketing Manager in Houston, Texas. It's my honor and privilege to introduce Dr. Stephen Pelech, a renowned professor at the University of British Columbia and the founder of Kinexus Bioinformatics and Kinetic Pharmaceuticals. Dr. Pelech, just a brief bio, received his bachelor's and PhD in biochemistry from University of British Columbia. Then he did his postdoc training at the University of Dundee with Sir Philip Cohen, and later on at the University of Washington with the famous Nobel Prize laureate, Dr. Edwin Krebs. He was one of the founding scientists of the Biomedical Research Center at UBC, a founder and president of Kinetic Pharmaceuticals, like I mentioned before. Kinexus has produced over 1,500 antibodies against cell regulatory proteins and deploys these in novel, high-throughput methods to monitor signaling proteins in biological specimens. Dr. Pelech has authored over 280 scientific publications in peer-reviewed journals and book chapters about cell communication systems implicated in pathology of cancer, diabetes, neurological, and immunology-related diseases. Dr. Pelech's talk today is on tracking and mapping signaling proteins and the SigNET KnowledgeBank. We're excited to learn more about this topic. Welcome, Dr. Pelech.
Thank you. It's a pleasure to be here, and I appreciate the opportunity from Signal Biologicals to present some of the work that we have here at Kinexus and to introduce a lot of the open access knowledge bases that we've created over the last 15 years with some new updates. So before I get too far, I should provide a conflict of interest statement. We are required to do this at the University of British Columbia, just to alert your audience that I will be talking about some of the services and products available through Kinexus, and my family owns about 74% of the stock of our company. So, and again, the presentation is really my views and not necessarily that of the University of British Columbia. So Kinexus itself is a spin-out company. We've been in business for over 26 years, and we're located in Vancouver, Canada, and not too far away from where the Signal Chem was located across the river, and that's facilitated a lot of our interactions with Signal Chem and now, of course, Cynobiologicals. And many of the founders of Signal Chem were actually former employees or students of mine in my academic lab. So there's a really strong connection over the years, spans more than 30 years with the people now at Cynobiologicals. So we provided over the course with this company proteomic services to, I guess, about 3,500 different investigators in both academic and industrial labs. And we've done this work for about over 35 countries. So that's just a little bit about the company itself. We also have the services we offer is an integrated platform for really biomarker discovery. We do everything from making synthetic peptides, including on membranes, through to doing Western blotting, producing antibodies, and basically doing research that's trying to map out the structure of these cell signaling pathways and forge these connections, and then document them in our proteomics databases that are available through the SigNET KnowledgeBank, which I'll talk about later. And this is very, as this audience is well aware, these signaling systems are really complex. Really what we've been endeavoring to do as scientists is to essentially reverse engineer this amazing technology in a sense that is in our fingertips, and to figure out how it is that we work. And then when we have pathologies, how can we actually assist the body in repairing that? And so I'm going to talk a little bit about the complexity of these signaling systems. And it turns out one of the big barriers to investigating them is making sure that your samples are prepared properly to preserve, for example, the state of phosphorylation of proteins inside cells, which is a really big problem, as I'll show you. And to really track these signaling proteins that are very low abundance, maybe 100 to 1,000 times lower levels than, say, metabolic pathway enzymes or structural proteins, you really need to have really good reagents. And this is where antibodies come into it. And we need to improve the throughput in analyzing a larger panel of proteins. And this is where antibody microarrays are pretty good. But a caveat is antibodies are not necessarily that specific, as we'll discuss. And finally, when you have these kind of technologies where you can get measurements of like 2,000 different proteins simultaneously from a single slide the size of a microscope slide, 1 by 3 inch slide, you got to be able to process that data and pull out and identify the rubies amongst the rubble, which is going to be valuable in providing understanding in your research program. And one of the things that we'll be introducing is the SigNET KnowledgeBank, which again is open access, and it has full of information that's been collected not just within Kinexus, but from the scientific literature. And we try to apply some predictive algorithms that can be useful for hypothesis generating. And so we'll talk about that. So in terms of the complexity of just the human proteome, this is the complement of all the phosphorylated proteins that might be present in a particular cell or tissue at a given moment in time in response to a particular perturbation or disease state. We know that the kinome encompasses, according to people like Tony Hunter, about 568 different protein kinases. Most of them are related to each other. There's at least 486 what we call typical protein kinases that feature a very common catalytic domain that catalyzes phosphorylation of proteins. And then we have atypical kinases. And then on the flip side, we have the enzymes that remove those phosphates from proteins, protein phosphatases, and there's about 159 protein phosphatase genes, and these can be divided into families as well. And so these are all converging to regulate the state of phosphorylation of proteins. And the number of actual phosphorylation sites that have been uncovered through mainly mass spectrometry studies is about 250,000 distinct sites in the human phosphoproteome. The actual number, however, is probably even 3 times that number, to 4 times that number. And there's a number of reasons why I come up with that number. I don't have time to go into that with this presentation, but I think it's pretty clear that most of that phosphorylation is actually irrelevant. You can call it junk phosphorylation, but there is very critical phosphorylation events that occur within signaling pathways. And the challenge is to sift through all of that and identify those kind of phosphorylations that may be actually functionally important in a cell. Now, mass spec, as I said, has been a very powerful tool to identify phosphorylation sites, but the main dilemma is that you have to enrich your samples first. And we have websites like Phosphocyte Plus, that's offered through Cell Signaling Technologies, and that was the founder of that. Dr. Peter Hornbeck used to actually be one of my employees in a pharma company. We both had a vision of trying to track phosphorylation sites and put them into a format where people could use it as a portal to find out more information about a specific protein and even specific phosphosites within it. And of course, CST's Phosphocyte Plus has many other forms of covalent modification they track as well. So it's a very powerful resource. And I think in combination with the PhosphoNet website, which I'm going to introduce to you a little bit later, together they actually complement each other. There's a little overlap, but they complement each other really well, and they're very valuable open access resources. Now, the problem with the mass spec, even though it's very good for discovery phosphorylation sites, it's very problematic in terms of tracking sites of interest. And this is illustrated, for example, that if you go to the Phosphosite Plus website and you look at the results of probably more than 40,000 or 4,000 plus mass spec experiments where proteins have been tracked for their phosphorylation and high throughput. If you focus on the 5,000 that are critical in signaling systems that have been well documented in the literature, about a quarter of them are not identified in high throughput studies. And another quarter have only been identified in maybe one or two studies. So there is limitations on the technology that many of the sites that you want to track that may be important in proteins are actually not going to be able to be identified by mass spec, largely because these are often highly phosphorylated, that is highly negatively charged. The fragments may be fairly short. It's not unusual for many of the serine-threonine phosphorylation sites to be flanked by arginine residues and lysine residues. And these, of course, are sites of cleavage of trypsin. And most of the data that's generated in mass spec is by trypsinolysis, and then looking at the peptides. So the other thing, too, is that when you're doing mass spec, as sensitive as it is, the reality is you have to first enrich your samples. And that enrichment process itself can introduce artifacts. Okay, and again, it's not very quantitative because unless you have some sort of internal standard, you get different levels of detection that are not necessarily fully reflective of the levels of those phosphopeptides in this case that you're tracking. So it has real limitations, but it's very useful. It is very expensive. It takes a lot of time. And often if you do a mass spec approach, and we've offered this kind of service at Kinexus with the University of British Columbia High Throughput Center for Biology, what we're finding is that you need to repeat the experiment multiple times because it's not always highly reproducible. And then even if you get the results, at the end of the day, you want to follow up with these fossil sites that you've identified. And really that requires the availability of antibodies to do it in a practical, cost-effective way. Now, using antibodies to track proteins, this is also highly dependent in the success of your experiments on whether or not those fossil sites are preserved after you've ripped open the cells and you've homogenized them and the contents are all mixed and you have phosphatase action, which usually have specific enzyme activities that are typically 10 to 50 times higher than you have for the kinases that are catalyzing the phosphorylations. So during the post-homogenization, you're actually dephosphorating your samples. And this actually results in an underestimation of the state of phosphorylation of many proteins. And this is despite the fact that we often are using phosphatase inhibitors in our buffers, things like sodium fluoride or sodium vanadate or beta-glycerol phosphate or EGTA, EDTA to chelate calcium. All of these different things help, but they're often insufficient. So our recommendation and what we do at Kinexus is that when we're studying these antibodies, we actually use them with lysates where the cells have been directly homogenized into 2 times concentrated SDS page sample buffer, and we get much better results. This is exemplified, for example, we take the A431 human cervical carcinoma cell line. These cells overexpress EGF receptors, so they're an ideal model to look at the regulation of the EGF receptor in downstream signaling events. And if we take 5 antibodies that we've developed at Kinexus, and we homogenize in buffers that include phosphatase inhibitors, and we keep everything cold, and we try to get to the SDS page step as soon as possible, the upper set of blots you see here is the detection after 1 or 5 minutes of EGF treatment of the cells that have been serum starved. And you can detect signals, but if you homogenize those cells directly in SDS sample buffer, you can see profound differences in the recovery and detection of phosphorylation of proteins. So, but the problem, of course, is that we like to also do work with microarrays, and we cannot use detergents like SDS, where we're looking at array format for the captured proteins, because the detergent's going to interfere with the antibody-antigen interactions. So what we do is we developed a method where we do chemical cleavage using this TSEP, tris-2-carboxylethyl phosphine hydrochloride, with a priest to reduce basically all the disulfide linkages, followed by treatment at an elevated temperature, or you could do room temperature for longer, with 2-nitro-thiocyanbenzoic acid, NTCB. And what this does is it cuts proteins essentially after cysteine residues. And these cysteine cleavaged, coupled cleavage peptides, we've calculated that if you take the entire human proteome and you do these treatments where the cysteine residues are, you're generating just short of about 250,000 different peptides. But these peptides, about, they're in the order of about 3 times the size of a tryptic peptide. So you're much more likely to capture a phosphocyte, plus the presence of arginines, like you would have again with the trypsin cleavage, doesn't become a problem in generating very short fragments that, for example, would fly through a mass spec. So there's a real advantage to this. And so what we normally do is we do a chemical cleavage, then we actually label these proteins with biotin. The biotin is binding to amine, free amines, so at the N-terminus of the peptides and on lysine residues. And this allows then the detection of captured proteins on our arrays, as illustrated here, where your antibody, or could be a dye-labeled antibody that recognizes biotin, or you can use avidin, which is dye-labeled, which will bind to the biotin that's on these peptides that are captured. And of course, there's a very high affinity interaction with these avidin biotin interactions. So this is how we actually can amplify the signals that we get when we're using arrays with the captured peptides. There's a number of other advantages. I mean, one of the problems with the whole use of microarray technology, and I think one of the reasons why it hasn't been as popular in the last decade, because it gives a lot of false positives and a lot of false negatives. And so in the ideal state, shown in panel A here, you're going to have your dye-labeled protein, in this case, captured, and the amount of dye signal is going to reflect the amount of protein that's captured. However, you can have scenarios where maybe another protein is associated with that protein and it may be captured as well. And if there's a change in the level of that associated protein, you'll see a change in the level of dye, and that may not be reflective of the actual target protein. You can also have scenarios where a totally different protein, because of antibody cross-reactivity, is binding. And another phenomenon which we came to appreciate is that the dyes that you use in your reporter reagents, whether it's with avidin or whether it's an antibody that you've dye labeled, or the protein directly, the problem is that the dyes themselves still, even when they're covalently attached, can have affinity for other proteins. And so the longer you incubate a microarray with your reporter system, the more likely that you'll have non-specific dye protein interactions. And because you've got concentrated proteins as antibodies on your arrays, your background signals go up with time. So we've learned that we do a longer incubation with your, in this case, biotinylated proteins with our arrays, but we do very short periods when we're now adding the dye labeled avidin, striptavidin. And finally, it could be that your epitope is blocked in your protein in the crude cell lysate that you're trying to capture it from, and it just can't interact with your capture antibody, and that's a false negative. Another thing that's a problem is when you have an intact protein and you're trying to monitor different the protein as well as different phosphocytes of that protein with phosphocyte-specific antibodies, is that your antibodies are competing for the same protein. So you may have internal competition on your arrays. So when you cut your protein into fragments, then you reduce that competition, you reduce the production of protein-protein interaction complexes, and you actually stabilize the protein because the chemical cleavage destroys the phosphatases and the kinases and proteases that might be at work to dephosphorylate or eliminate your proteins that you're trying to detect in the first place. So this is a strategy that we use, and it's very effective. This is an example here, for example, of one of our antibody microarrays, the CAM220 chip. We have at least 2,026 antibodies on this array, each printed in quadruplicate. So you have over 8,500 features on the one slide. And of those antibodies, of the approximately 226 antibodies, almost half of them are phosphocyte-specific antibodies. At least that is the intention. And these are targeting these low abundance proteins that are regulatory, that we think are important in these cell signaling pathways. Well, again, antibodies are not necessarily specific. And I think the worst antibodies for specificity are phosphotyrosine site-specific antibodies. We've actually produced about 1,600 different antibodies in-house against, most of them against phosphorylation sites. And we observe with the tyrosine phosphorylation site antibodies that they're often non-specific generic phosphotyrosine antibodies. And so it's really important, and we do this in our purification procedures, to do negative purification as well against phosphotyrosine. And through that, we've actually produced a very large amount of generic rabbit polyclonal anti-phosphotyrosine antibody. We call it PYK antibody, which is very, very inexpensive from the company and actually has a broader range of detection of tyrosine phosphated proteins than, for example, the monoclonals like 4G10 or PY100 and the PY1000, for example, that's currently being offered. The polyclonal preps are better. But anyways, back to the intended phosphocyte-specific antibodies, we've invested a lot of work in using our spot array technology, where we can make these peptides on arrays, custom design, and then we can test our antibodies against different phosphopeptides that are very similar in their sequences to get a sense of how specific are these antibodies. Here's an antibody that was developed against the cyclin-dependent kinase 1 threonine 161 activation phosphocyte. And you can see the sequence of the peptide there. And we have an array that features about 30 different phosphopeptides on it, including, as boxed with the red box, the B2, which is the target that's the peptide that we wanted to have antibody response to. And you can see that it'll have some cross-reactivity against what's shown as the C5 spot here, which is a CDK7 phosphocyte, which, as it turns out, has a very similar sequence. So there's always this risk when you're using phospho antibodies that related sequences may, in fact, be picked up with your antibodies. And if you've got, as we believe, about 1 million phosphorylation sites that exist, it's inevitable that you're going to get some kind of cross-reactivities happening. And when you look at a lot of the fossil site antibodies that are being offered by companies, usually you're seeing, you know, one or two spots under the most ideal conditions where they appear to be very specific, and hopefully they give a strong detection of the target signal. But in fact, when you look at a wider range of tissues with that same antibody, you can often find other proteins that are reacting even much stronger. So one of the things that we do at Kinexus, and we have this on our products website where these antibodies are offered, we have information for pretty close to 6,000 immunoblots and dot blots, some including like what I'm just showing here, of the characterization of our antibodies. So you see the good, bad, and the ugly. Sometimes those other cross-reactive proteins may in fact be very useful because they show you remarkable changes in the model system that you're looking at. One of the other things that we have done is to see how specific are these for the fossil sites. And so here we take the same antibody and we make a series of peptides, each as individual spots in the top panel, and wild type corresponds to the original sequence as shown above. And what we do is we do substitutions of the fossil site, the phosphothreonine in this case, with the phosphocserine or phosphotyrosine, and then the unphosphorylated version. And we also do glutamic acid substitutions because being very acidic, there's a number of phospho antibodies that will actually pick up just glutamic acid or aspartic acid. So the panel below shows you our control where we're probing just with the secondary antibody. And as you can see, this particular antibody picks up phosphoserine just as well as it picks up phosphothreonine. But in fact, it'll also weakly pick up phosphotyrosine. So we've done this for about 3/4 of all of our phospho site antibodies. And that information is documented on our product's website. Now, when you put these antibodies on the microarrays, and here the beauty of these microarrays is that you have a very concentrated antibody in a small space. So as long as you have a good detection system, and the more of the protein that's there, the more detection we have, and it goes red. And it goes from, like, as it gets less protein, it goes red, orange, yellow, green, blue, and then you don't see anything. So we get a pretty good quantitation, and we get a dynamic range that's about 2,000-fold. And when we look at our duplicates, our mean value in the variance from our duplicates is about 5 to 6% from the two measurements. So it's actually pretty good in terms of its reproducibility as documented on the same slide with the same sample. And what I boxed here on this example that's shown here is where you can see that pre-treatment of the sample with, in this case, alkaline phosphatase, which should dephosphorylate proteins that are in the lysate prior to incubation with the array, you can see this reduction in signals that occurs, again, showing you clearly that you can detect these phosphorylation changes on these arrays. And so you can do this with in vitro studies, like we just did with the phosphatase treatment. But of course, when you take cells and you're treating them in culture or with different treatments, or you're taking diseased tissue from animal models or whatever, here we just focus in on part of the array where in these, again, A431 cells that were treated with EGF, we can compare serum starved cells in the bottom panel with cells treated with EGF. And again, this is only 5-minute treatment, just focusing in on one part of the array. And as documented here with a lot of EGF phosphosites and some other proteins, you can see these very profound changes in phosphorylation. So this method actually is pretty powerful. Now, what we've been doing more recently is taking our microarrays that have over 1,000 phosphocyte antibodies, and we've been preparing libraries of peptides. So we take a mixture of different, in this case, tissues from mice tissues, So the objective here is to get a representation of the human proteome, or sorry, the mouse proteome. We could do this with obviously human tissues or human cell lines. And then you can treat with a phosphatase to remove the phosphorylation sites further, but most of the time, if they're unstimulated, most phosphorylation sites are rapidly lost. So you create a library that with the chemical cleavage step, it represents basically the human, in this case, mouse proteome. And now you take a purified, and remember, the treatment with the chemical cleavage destroys the endogenous kinases and phosphatase activities and protease activities. These samples, once they're subjected to chemical cleavage, the phosphates are stable for days at room temperature on the bench. So this is really a great way of preparing samples that can be shipped to other places. Doesn't matter. So but with a dephosphorylated library of peptides, you can now take, for example, a purified protein kinase, and SigNET Biologicals offers very active preparations of protein kinases and phosphatases, and or you could, if you wanted to, like I say, you can take these purified enzymes and then incubate them with the library with ATP. So then you would be phosphorylating peptides that are really natural proteins with a huge diverse library. And then you use the incubation afterwards with the antibody microarray to capture the phosphorylated peptides. And you compare the results from that analysis with the results when you've just added ATP to your library and captured the microarray. And then you get a large number of hits. And then what we find, though, is we do get interactions that are sometimes not expected. So we apply a filter. And our filter is to use basically our predictive algorithms for the specificity of these kinases. And I'll come into that, explain that in a moment. Where with the application of this filter, we can see if the hits that we get from the microarray make sense for what we know about the specificity of these kinases. And then we generate a list of hits. So here's an example of some of the hits from taking cyclin-dependent kinase 5 and ATP and incubating this library of mouse proteins, and then looking at our top hits. These would be the signals in the first column with just ATP, and in the second column here, this is the intensity of the signal is increased. This is the percent change from the control. That's the signal increase. Now you may wonder, well, why is the signal not so dramatic? Don't forget that we do have a background phosphorylation for some of these proteins maybe already in the original lysates. Plus, we do still have this issue of a little bit of non-specific dye binding. But here we were able to do statistical analysis that these were using a student t-test, that these were all increased with the addition of the CDK5. And then in the blue column, you can see the actual sequences of the peptides that were used to make the antibodies. In the blue column, this is the prediction with our SigNET kinase-substrate predictor algorithm that ranks the kinases in terms of the prediction for their ability to phosphorylate that sequence that's found there. And so the best would be a number 1. And what SigNET website gives you is the top 50 out of 500 scores and ranks them as well. And so this is showing that these observed phosphorylations using the antibody microarray are also predicted to be within the top 50 most likely kinases, the top 10% of kinases that would likely phosphorylate those sites. So I think this is a good way to discover brand new sites that have never been identified before for particular kinases or phosphatases. Shown here in the first column, in the slightly darker green, is the P70S6 kinase. That was a confirmed phosphorylation site in the literature for being phosphorylated by CDK5, but none of the other sites have ever been identified before in the literature as CDK5 sites. And of course, using the Western blotting approach, you can also try to add to these cell lysates, not hydrolyzed, but with these kind of clues that these proteins could be substrates, in this case, again, for CDK5, we can then take and add ATP and the CDK5, the recombinant enzyme from, say, SigNET Sinobiological, and we can then look to see on a Western blot, does it actually show an increase in the protein that's of the expected size, and confirm these phosphorylation sites. So it's very enabling. You identify antibodies that you can then go back and identify brand new substrates for kinases of interest. And so we kind of like have a platform then where we discover these kind of phosphoproteins that are changing both in vivo or in vitro with these kinase experiments. We can do multi-blotting where we can do a Western blot to see, well, is the antibody signal from the arrays corresponding to the target protein, or is it a cross-reactive protein that in fact is demonstrating these changes? And then once you establish the specificity of an antibody in a given model system, then you can take and create macroarrays or microarrays with those lysates from these samples and spot them as individual, maybe in triplicate as shown here, samples, and then probe them with an antibody. So this is a reverse lysate array. Unlike the microarrays where you have the antibodies arrayed out and you're probing with a lysate, here you have all these different lysates, and now you're probing with a particular antibody. Okay, so we do have as one of our knowledge bases the SigNET AM for antibody microarray website, where we've got the results from about 2,000 microarray experiments that we had done, about 1.5 million measurements. That's tracking about 700 different signaling proteins and their phosphosites. We haven't got the information from the last 5 years in this database, but we intend to upgrade it in the near future to include the results from another 2,000 microarrays. Now, the SigNET AM database here is one. Again, this is an open access knowledge base. You can screen it through a treatment and find out what proteins have been affected, or you can do it through a protein of interest and find out what treatments affected. You can query it in that way. Now, we have all these other knowledge bases that we've created. And one of the first ones was PhosphoNet, which I've alluded to earlier, which is a database of human phosphorylation sites. And I'll come to some of these other knowledge bases as we discuss this in the last half of my talk. So the PhosphoNet website has about, at the time that we made it, there was about 177,000 confirmed by mass spec phosphosites. And we collected this information from Phosphosite Plus and other phosphorylation websites at the time. And we used that information in part to actually educate the algorithms that we prepared for prediction of phosphorylation sites. And we came up between the known sites and what we expect to be predicted, something close to 960,000 known or predicted human phosphorylation sites across about 90% plus of the proteins encoded by the human genome. And just to give you an idea, I mean, our knowledge of the number of sites in terms of their regulation is really, we're just scratching the surface. You know, maybe at this point, 5% of the fossil sites, we have some idea what they might be doing in terms of the regulation of a protein's activity or location or degradation. So what we did was, with using both the known and the predicted sites, we started to also take data that we had for known protein kinases and their known substrates, and use that information to train algorithms to recognize phosphorylation sites in proteins that were not previously shown to be substrates of kinases. So in addition, we were very interested in seeing, well, if you have a protein that features a phosphosite and you know a kinase phosphorylates it, and another protein has a very similar site, then maybe that other protein might be potentially phosphorylated by that kinase. So we started to try to match the relatedness of the phosphosites to each other. And then we were very interested in trying to identify functionally important phosphorylation sites. We wanted to know, well, if a protein phosphorylation site found in humans is found in other species, right down through into yeast and red bread mold, maybe it's functionally important. So we're very interested in the evolutionary conservation of phosphorylation sites. And then, of course, we wanted to link to other information about these phosphosites. So this is sort of like, if you went to the PhosphoNet website, and by the way, the main Kinexus website is www.kinexus.ca. We're based in Canada, so CA at the end. From that website, you can go and find all these other websites. There's direct links there. Now, the Kinexus website is just being updated. So we hope next week to launch our brand new Kinexus website that identifies a lot of these new products and our services, but also the links to these websites. And in about a month, we should also be updating our products website, our Shopify website. And the reason why the Shopify website is really of value to you as a researcher, even if you don't buy our products, is that all of our phosphoantibodies and pan-specific antibodies are documented there, and the results from all of our internal testing is on that website. So as I said earlier, you can see our Western blotting in different mouse tissues, or our Western blotting in different human cancer cell lines subjected to different treatments, or our spot arrays, where we're now looking at these protein phosphorylation sites with slight differences, or even some of the purified preparations that we've obtained from SigNEL over the last decade, where we then have been able to test the purified proteins against these sites, antibodies, and pan-specific antibodies. I mean, this is again the challenge, is that you may actually have a good antibody, but when you're preparing your cell lysates, you may not have the right model system for that particular phosphosite to be maximally phosphorylated so you can detect it with the antibody. So the antibody may actually work, but it's just not being tested properly. Or vice versa, again, you may have an antibody that's giving a strong signal, but it's not your target protein. So anyways, you do a query on the PhosphoNet website. You can query by ID number or the name of the protein, aliases, and then it'll pull down a menu. And on that menu, you'll see these columns that gives you the phosphorylation sites at the bottom. There's links to other websites here as well. And these columns, if it's green, it's predicted. If it's white in the first column, then that's a confirmed site. You click on it, it gives you the reference. If there's a known effect, it's indicated in white. You click on it, and it puts it into the box that you can see above there, the text box. You can copy and paste that. If it's a known kinase that phosphorylates it, it'll be listed. If it's a known phosphatase that dephosphorates that site, it's listed. And you'll have all of your phosphosites listed for that protein. Now, in the orange buttons, the first column gives you a link to the paper where that phosphorylation was described. And then the second column gives you evolutionary analysis. The next column to the last column is kinase prediction. And the very last column is phosphocyte matching. So when you do the evolutionary analysis, it'll take, in this case, an AKT phosphorylation site, threonine 308, and it'll line up that sequence where the algorithm is looked, found, retrieved the homolog that's for that, or the cognate protein in that different species. It generally aligns pretty good. Sometimes it's out by a couple of residues, but you can tell that when you look at the output. And it'll show you if that site's highly conserved. And actually, it'll further down calculate the percent amino acid identity of the whole protein and of the phosphocyte in these different species. Now, the phosphocyte match will take that phosphocyte, in this case, that threonine 308 site, and it'll look to see all these other proteins that have almost the same amino acid sequence. So let's say you have a cross-reactive protein that's showing up with this antibody. This can give you some clues to what that protein might be, because it shows you not just the sequence, but the size of that protein, and you can see how that matches on your SDS page gel. And then finally, that kinase predictor, it gives you a score for each protein kinase for whether that sequence, that 15 amino acid sequence, is optimal for the consensus sequence of what that kinase recognizes. And we have consensus data for about 200 kinases from actual experimental data, from the literature, from our own use of synthetic peptides as substrates, and then adding that information in our training. And then we use that training to then predict the other protein kinases, about 500. And you might notice that we have what's called the max score here, the first entry, and then the other scores. And we can use this kind of information to identify a subset of phosphocytes here, which have a very strong high score with a particular kinase, but because the sum of all the scores is relatively low, it's much more specifically phosphorylated by that particular kinase. So this is one way of honing in these phosphocytes on what might be very important functional phosphorylation sites in cell signaling systems. So in addition to having PhosphoNet to identify phosphocytes, we have KinaseNet, which has very detailed information about each of the protein kinases, including, again, their evolutionary conservation, their primary and tertiary structures, compounds that are known to inhibit sites of phosphorylation that regulate that kinase, substrates of that kinase. Here is the typical output that you'll get when you enter the name of a kinase or UniProt ID. Again, these are all human protein kinases that are being featured here. But in each of them, we have a matrix. And the matrix is generated from the testing of all these substrates. So you may see many, for example, the logo diagram has been used to describe how frequent certain amino acids are located around the phosphorylation site of a particular kinase, or even for an antibody specificity. We instead create matrices, and these matrices have all of the 20 common amino acids and all the positions surrounding the phosphosite, the 0 position being the phosphoacceptor site. And this gives you a quantitative matrix that takes into account also the expected frequency of these amino acids on the surface of the protein where the phosphosite is located. So if you get a negative score, it means that that site is less phosphorylated in substrates than you would expect just by random. But if it's a positive score, then this site seems to be that particular amino acid at that amino acid residue position is a very favored. And so we use this information with a scoring matrix to do all 970,000 known and predicted phosphosites that's in PhosphoNet. And to show you the power of these predictive algorithms. Here we have consensus sequences that are determined empirically from experimental data where the substrates have been all aligned and we get a consensus sequence. If we use our prediction algorithms, and we do this for the yeast protein kinases. So this is experimental data. It was from a yeast study done with about half of the yeast protein kinases. And we do the same with our prediction algorithm using the amino acid sequence of the yeast protein kinases, nothing else. We get this as our predictions. And as you can see, we get pretty nice matches, including the importance of glutamine residues, or tryptophan residues in recognition. That is not common with more of the multicellular eukaryotic kinases. So it's pretty powerful. Finally, we've also introduced a couple of the other knowledge bases. We have TranscriptoNet, which has gone through the GEO database, Gene Expression Omnibus. We collected all the information that we could from DNA array studies for gene expression levels in response to different human organs and different treatments. And so we then took that large database, normalized the data, and now you can actually find out which organs have which proteins, at least predicted based on mRNA expression levels. And we've identified what are the most common genes, what are the most unique genes for a tissue type, and also those genes that are differentially regulated, for example, in human cancers. We have a large number of 60 different types of cancers where we've documented this. Again, it's all open access. And then for the cancers, we've taken all of the about 3,000 proteins that have been linked to cancer and documented them in terms of their mutation patterns using a lot of the information that was originally provided in the COSMIC database from the Wellcome Trust. And we've again normalized that to identify what are the most common mutations that are in these oncoproteins and allowing discovery in part of what might be other proteins that have not yet been identified as oncogenes, but based on mutation patterns, we would predict that they might be. So that's also of open access. And another knowledge base that I think will be extremely useful, especially for teaching purposes, and maybe just getting a sense quickly when you're doing your own studies, what's known. We've documented about 22,000 kinase-substrate connections at Kinexus, and we have more that we're adding to this. And the objective here was to try to put these into maps that you could actually track cell signaling. So we have direct maps and we have indirect maps. And I'll explain also a new module that we're adding to look at the known extracellular mediators. What receptors do they bind to on cells? And then those receptors, what proteins do they directly bind after the engagement of the extracellular mediator, its ligand? So that's a new module that will be in Connector shortly. I have a team just finishing that off of computer science students, and we're expecting to have that launched by the end of this month. So watch out for that, and I'll explain a bit more about that in a moment. But when you go to Connector, at the top, you can do queries where you pick a protein, and you can identify all the kinases that phosphorate it, or you can pick a kinase, identify all the proteins that are phosphorylated by that kinase. And you can customize the map that you generate with these buttons. And then you get an output. And the output kind of looks like this, where you've got your target protein that's of query, and you've got all these proteins that are known substrates of that kinase. And they're color-coded for the class of the proteins. And when you follow the lines, the sites that are phosphorylated on that protein light up. If you have more than 3, then that protein's repeated on the next line. So what ends up happening is when you run out of proteins, then you click above here. And so, for example, there's 28 pages of maps. for substrates of AKT that when you just click on this, it'll take you through the entire map, and it's alphabetical. So you can tell how far you need to go to get to the protein that you're interested in based on its name. And if you click on the protein, then all of this pop-up appears. And from there, you can link to all these different websites with detailed information on that protein. Both our SigNET knowledge bases and other reputable websites from other government agencies or PhosphoNet. Now that's if it's a direct interaction, but we've also have a equivalent of kind of like MapQuest that you can start with a kinase and you want to know, here's a protein that's undergoing an increase in phosphorylation. Knowing all the known connections between kinases and their substrates, which include other kinases, you can actually then track how it is, for example, in this case, how the ELK1 transcription factor actually is targeted by RAF1 and what could be intermediate kinases, and there could be multiple routes. We have about up to 5 degrees of connectivity that we're tracking here that you can query. And when you click on the line, the phosphosites light up. And then when you click on the phosphosites, it takes you to other websites, Phosphosite Plus or PhosphoNet, with detailed information on those phosphosites. Now, a new feature that we're adding to the connector website is a human body map. And so in this body map, what we've done is we've taken about 60 tissues and cells types, and we've coded them to try to show them where they're located, including regions of the brain. So they all have this light gray kind of background in the map. And then here I'm showing you the location of the estrogen receptor and the location of the testosterone and androgen receptor. And so if a tissue or cell has that protein present at reasonably high levels, then the organ or tissue or cell colorizes, and the gray text that identifies it goes bright green and has a bright green line that connects to where it's located. So this is being done to show you the locations of all the extracellular mediators. If it's a small molecule, we identify where the enzyme is that produces this small molecule, and where all the receptors are to accept those particular ligands. And when you pick an extracellular mediator, it also shows you a table of all the proteins that where the receptors are, the cell tissues, for that particular mediator. And if you pick a receptor, it shows you all the mediators and where that mediator is found. And you can have multiple mediators for a particular receptor, and you can have multiple receptors for a particular mediator. So anyways, this is kind of a cool feature. It's something that you can output all of these maps. You can download them as PDFs after you've customized them. So again, this could be very useful for teaching. Finally, I'd like to briefly introduce you to the drug KINET. Those of us that are engaged in mapping these cell signaling systems, really it's to identify drug targets. And kinases, probably about at least a quarter to a third of all drug development today is protein kinases. So we created the drug KINET website to document the known interactions of drugs with kinases. In many cases, certain kinases are not sensitive to a drug. We've recorded that too. And we developed algorithms that gives you empirical data on the sensitivity of all these different kinases to the drugs, and also insensitivities, and also predictions. Now, one of the things that's powerful as one of the modules of drug KINET, and you can do this query, you can pick a drug, and you can get the kinase that has a 3D X-ray crystallographic structure with that drug, or you can go the other way around. You can do it with the kinase, find the drug, and then get that pair, drug kinase. And you'll get the structure of the drug with the atoms numbered, and you'll get the structure of the kinase and where the drug fits into the kinase. And what this algorithm does is we've calculated the bond distances between every atom of the drug with every atom on the kinase target. So each of the different side chains that are in the vicinity of the drug, we measure the bond distances between each of these atoms. And we are focused on drug atoms and side chain atom interactions. You can vary how many nanometers that you want to go for that interaction to be recorded. But ultimately, you generate a table that the bottom table here lists every atom interaction between the drug and the kinase and the bond distance and the number of times that interaction, there's separate interactions of documents. So you have hundreds of potential atom interactions of a drug with the kinase. Then the top table filters that information and it calculates the number of times that side chain is interacting with the drug and the average bond distance of each of those interactions. So the closer the atoms are, we think the tighter and more important the interaction. And the more frequent that those side chains, that particular amino acid is interacting with the drug, we think it's more important. So you take the total number of interactions, divide by the average bond distance, and you get a ratio. And that identifies the most critical amino acids for the binding of that drug. So you can actually predict with what kind of amino acids are going to work. Now, you can work this backwards, and there's a feature that you can start with the genes. You can start with what you think is the critical amino acids, and it'll go back and identify the SNPs, single nucleotide polymorphisms, that are likely to actually reduce the ability of that drug to bind to that kinase. So if a person has a cancer, for example, and they're taking a drug, and you get their genome sequence, you can actually work from the identification of the drug interactions to find the SNP. So if a person has that SNP, they're not going to be as susceptible to the drug. So anyways, this is all open access. It's on the website. And finally, everything that we did for the drug KINET website, we created drug ProNet. So it does the same thing I just described to you, but now it does it for about 480 different proteins based on X-ray crystallographic data with their interactions with over 2,000 different drugs. And that includes also the protein kinases. And so now you can actually expand this to looking at drug action in general. So that's my presentation. I know we covered a lot of material. I hope I've gone too much over time. I do acknowledge that this can only be done with the help of so many other people, including people in my academic lab and at Kinexus. And I'll point out that a lot of the retrieval of the information that's informed to create our databases has been done in part through the help of students in my graduate courses at the University of British Columbia in my MIDI 590 class that I teach. And the creation of these websites, that has been possible through teams of students, of computer science students from the BC Institute of Technology, where they have projects. And so each of these knowledge bases reflects the work of a lot of annotation from these students from the university, and a lot of the programming and development of these websites from these BCIT students. So thank you so much for tuning in. And I hope that there's something here that has been useful in going forward. And we'd love to hear from you at Kinexus.